Welcome to another enterprise session from the University of Bristol. I'm Professor Michele Barbour, and today I'm joined by not one but two subjects, interviewees, victims, I don't know. I have with me Professor Ash Toye and Professor Jan Frayne, both of the School of Biochemistry. Thank you very much for joining me. I know you're both individually very busy, and trying to get you in the same place at the same time, I really appreciate you finding the time. So thank you.
No worries.
Thank you for inviting us.
I'm really looking forward to hearing your story about your research, your entrepreneurial journey, your spin out, Scarlet Therapeutics. But I also need to point out that usually I interview one person at a time. And I'm really fascinated by what will emerge about the dynamic between the two of you as sort of joint academic founders and colleagues, and however you see yourselves. I'd like to have that at the forefront of our discussions as well, because a lot of the people I talk to are solo entrepreneurs and you guys aren't.
Kind of strange that people would choose to do it on their own, I think. In my head, I thought it actually feels a bit safer.
I think sometimes it's finding someone that actually has enough aligned interests and expertise and aspirations as yourself. That's why I did it on my own. It wasn't because I wanted to, it's because there wasn't really anyone else around me who was in the right place to come on that journey with me. So it's necessity more than anything else.
I guess you've got to have enough enthusiasm for the topic. And so the lucky thing is we both work in the same area and we've both collaborated, right?
Yeah, I think we were collaborating anyway. We're working in the same area, but with our own distinctive part of that area. And I think when it came to the company, we just both happened to have something separate that then came together to make the company.
Brilliant. Let's get to that, but let's do this in a linear form. So let's rewind, first of all, if we can. You've both been here at the University of Bristol for a time, but I'd love to hear a little bit about your backgrounds. You can take that back as far as you like, but what were you doing before you came to Bristol and what brought you here?
Gosh, before I came to Bristol, I did my first research job at Cambridge University, straight to my degree, and then I was offered a PhD. So I came to Bristol for the PhD, and like many people, haven't left Bristol.
It's very sticky.
Ever since then. My research initially was more in the reproductive area. So I was working sort of on sperm function, and then slowly I moved into erythropoiesis.
The dark side.
Very much helped by a wonderful man, Professor Dave Anstey, who's not with us anymore. He was at NHSBT, became very much a mentor, and then slowly just immersed myself into the area of erythropoiesis. There are some similarities I can draw
just before we go,
but,
what is that long word you just said?
Erythropoiesis, the development or the creation of the red blood cells in the body. Yes, so we work on the development of red blood cells and red blood cell diseases, how the process of making red blood cells, which take place in your bone marrow, is actually regulated. So it's a very dynamic process. You're making about 1 million red blood cells. They're churning out your bone marrow every second. So,
wow.
2 million.
Yeah. 2 million, sorry. I cut out that. If you were anemic.
2 million per second.
I'm starting to get the dynamic between the two of you. But that's quite a leap, though, from like sperm function and motility and so on. Was it difficult making that transition?
I didn't want to leave it behind, so it was sort of a gradual merging process along the way. In some ways, studying sperm function is difficult. It's not an easily fundable area. Things like contraception's not massively fundable because you can't have any side effects, essentially. But when it comes to techniques and you're thinking about cells without a nucleus or with a condensed sperm have a very sort of condensed nucleus. Sperm at the end of the day is just this transport system to carry the male DNA to the egg. Red blood cells, in my mind, have some similarity because as they're developing in your bone marrow from a stem cell, they lose their nucleus. And it's also a very, very dynamic process. So the techniques, I mean, with lots of research, the techniques you use.
Yes, I guess so. Lots of transferable skills and insights and so on. Yeah, okay, I can see the link much more clearly now.
And I think also with the red blood cell field in Bristol, we had fantastic people. It's a great place to be doing this sort of research. And I've got Ash here. Ash actually was also linked with NHSBT, as I'm sure he'll tell us in a minute. But we had a big team up there to work with as well. So of all the places in this country to work, red blood cell research, we're lucky.
So the environment was inspiring as well as the actual subject.
Yes.
So, yeah.
Bristol is the red cell corner of the UK.
I mean, Bristol has many epithets. That's not one I've come across before. I shall adopt that. I like that. Yeah.
So it's got one of the biggest. So the blood service here is one of the biggest in Europe.
I knew that. Yes. Good point. So what about your background, Ash, before you were in Bristol?
So born in Wales, and then I came over as an undergrad student to biochemistry, and then they locked me in biochemistry until now. So I've done the whole pathway. So I've done undergrad, PhD. My PhD was on Alzheimer's disease, postdocs. I got a fellowship, so became self-funded, and then took that forward, become a lecturer, senior lecturer, reader, professor. I've seen the whole thing. I've seen the school
the
change
whole spectrum.
Over the whole...
This makes Jan and I, as people who came here for our PhDs, look like new girls, like blow-ins.
Outsiders.
Exactly,
Yeah,
absolutely.
Well, I did leave Wales, so that's...
yes, but
and it
Bristol
wasn't, in my defence, at the time it was really unusual. Like I came from, not a super rich background or anything. Nobody had gone to university as far as I'm aware in my family. But I just had this urge to do science. I don't know what it is. And originally I was going to be a farmer.
Sorry, pharma with an ER or pharma as in pharmaceutical?
Farmer as
in animals or crops. Or, yeah.
Do you know what? I didn't know that. I mean, another link we have, we're both Welsh. I'm from Cardiff as well.
Yeah, so I'm from Caerphilly.
Yeah. And I'd always had this decision that maybe by 28, 30, I would become a sheep farmer. That was and I worked on sheep farms. And we're discovering this live.
So I actually So I worked on the farm for all through my teenage years. So I didn't know this.
You were natural scientific soulmates all along,
But then luckily I remember literally being, I think it was a Cardiff University person who came to visit schools, like, careers day. And they said, come and do biochemistry. And I looked up what biochemistry was. And I had a biology teacher, actually, so who had done biochemistry. And I was like, you know what, this sounds just right. Because it was like how cells work, all the bits inside a cell. And then I applied and then I got into Bristol. So, and then it went from there.
It's a little known fact that until about a week before the university application deadline, I was doing biochemistry too. And I pivoted with about 3 days to go to physics. I'd read a book on the discovery of the double helix, which is sort of biochemistry. I read a book because I thought it would be relevant to my university applications. And in it was a little bit about some X-ray crystallography and some techniques they'd used, which were very much physics techniques being applied to biological systems. And I realised in the entire book, the one bit I could really remember was the physics techniques, not all the biology, chemistry, biochemistry, and so on.
Yeah, so often it goes the other way. It's physics people going into...
I think that direction is more common, isn't it? And in some senses, I've done that since.
And we need those links so much more now.
We do, absolutely. Yeah. So that's what brought you to Bristol, brought you to your roles today, and you're both involved in, I'm not going to try and use the formal word, we're going to say red blood cell research. This is a great environment, as you said, to do this kind of research. You could quite happily have remained researchers for your whole careers. You didn't need to go and form a spin-out company, presumably. Was it your aspiration to do something entrepreneurial, or did it arise as a solution to a problem or a challenge you encountered? How did it come about?
I imagine it's going to be different for both of us.
Great, well, let's hear from both,
It was like a whole bunch of different things. So one is having been in Bristol for so long, in the same building, going through the same, well, not always the same door, but most of the time, it's kind of looking for something different. Like it feels, sometimes academia can feel a bit like, I guess, a hamster wheel, right? Because you get funding, you get students coming in and you have postdocs and you work on something exciting for a bit and then you write a paper and then you might carry on or you might go off in a different direction. And that's exciting and really cool. But when you've done it for a period of time, it's kind of like, is there something else?
Yeah.
And you keep hearing on the periphery of people potentially spinning out companies. And it's always something that's been in my mind. But also alongside this, we had a collaboration with the Blood Service and we were trying to do a clinical trial, and it took about 10 years and we're nearly finished it now. And that was on lab-grown blood. And I realized that there's no way you can do this in academia. You can maybe do one clinical trial, but then it's got to go into a company to go faster.
The sorts of funding that universities can attract is not the sort of funding that would be required.
And the same for the Blood Service. The Blood Service obviously was like, well, we can't keep funding this forever. So at some point you realise, well, if it's fundable and could potentially be in a company, then we should try and do it. And actually, I approached the university way before I had that decision about another element. And I was like, can we patent this and can we do a spin-out company? Because I was interested in it. And they were like, nah. So I actually had that from the university originally and it put me off. And then what really annoyed me and kind of what spurred me on was a company was formed in America on the same idea. About 6 months later, they got a £50 million investment. And then they got £450 million. And then it got floated on the stock exchange in America. And then it went up to 2 billion. And so that made me think, hang on a minute, I've watched this story. I literally, as soon as they got the 50 million, I was like, see, this was a good idea. And they admitted that they missed something. But then I'd never let go of it. And sometimes having somebody else with you to go on the journey is also fun as well. So
we'll come back to what the company's actually doing, because I really, really want to explore that. But your motivation is a combination of, I'm going to put it in my words, because this is similar to my story actually, is I've been doing a similar job for a time now and I actually need something a little bit fresh to infuse it with some energy. But also the wish to move your technology forward and recognising that there's only so far I can go in a university setting. You need different forms of money, support and so on.
Yeah, and also there's kind of a third element, which is we have really good people come through the lab and you train them up and some stay for quite a long time. Well, then what do they do? And so it was also trying to like harness and give another career route for them. And then there was some I could always see coming through who had an interest in being an entrepreneur. So it's kind of like seeing another element that could be utilized.
I do think that's so powerful for early career researchers or postgraduates, whatever we're going to call them, because we often paint academia as the route, and it's not. It's a route. It's one of many routes, and it's not right for everyone, and it's not
really
even the ones that want it won't all get it. So it's so important to show that if they can see it, they can be it from your point of view. Jan, then you've got different motivations for the spin out or different context?
I mean, the whole spin-out idea was Ash's idea.
Yeah.
And you had to drag me in, kicking and screaming.
Well, you didn't resist too hard.
No, I didn't resist too hard. Because I suppose in many ways I'm so immersed in my university academic research, but we had the technology to actually take it out. So it's been really, really good fun, but a completely different world and language.
Jan, Ash was already keen to form a spin-out company and so particularly motivated, but you said he had to drag you kicking and screaming. Was it that you were reluctant or just was sort of, well, I'm busy, I've got my things to do, I don't really need another thing? Was there actual relucance or just
I think just the unknown.
Yeah.
I mean, really, really unknown. I mean, Ash had been thinking about this for a while and had
Slightly
done some
obsessed.
exploration of it thing. So the whole concept was completely new to me. So yes, then we started going to meetings, but it was a completely new world, the language, everything. I knew nothing about it whatsoever. So it was quite a big learning curve, but hugely supported. So it's been great,
brilliant,
but completely different.
You say obsession, I mean, we're researchers. I don't think you can be a researcher if you're not obsessed.
No,
You need to be a bit obsessed with something to be able to properly research it.
Well, people ask you, right, what is it like being an academic? And it's like, well, doing something you really love for your entire career and going in any direction. The only thing you have to do that maybe you might not like as much is sometimes there's difficult periods when you don't get funded and things like that, right? Or there's issues with students or whatever. But on the whole, it's exciting. And sometimes you're doing something that nobody's ever done before, right? And when somebody shows you that blot, or I remember literally the first time we produced blood in the lab, and we had a tube of blood. And I remember the postdoc at the time, Emil, like standing there in the middle of the lab and going, here it is. Like some blood. It was just in an Eppendorf. It was a really small amount. But at that moment, I knew that we had something that other people didn't have.
It's a hair stand on end sort of moment,
it's
isn't
like
it?
a pivotal moment, I guess. And then you're like, what could we do with that?
So tell me, zooming out a little bit, it's kind of obvious why we might want lab-grown blood, but tell me why did you try and grow lab-grown blood, and how, in the simplest terms you can express it, how does one go about that? Because there's all kinds of factors I would imagine that you need to take into account.
So similar with academia, right? You pick something and you study it. So at the time, if I looked at all my career, a lot of my career has been around model systems, right? So you, like, if you work on Alzheimer's disease, you need neurons in a test tube to study. We wanted to study disease originally, so different types of disease that affect red cells. But when a red cell's made, it's made and it's floating around your body, and when you access it, you can't study anything because it's just a red cell. So we realized that you have to make a red cell to study how it's made, and that's where then we began to understand how red cells are made, and then we realized we can manipulate them and what goes wrong with disease, and then study disease. And then it's like, well, actually you can make therapeutics with them.
But I love it. So your initial intention wasn't to make red blood cells to make the blood. It was, but so that you could then use it for research on the actual formation.
Originally the motivator wasn't to treat people in the clinic or anything. It was to understand where eventually things we studied might have turned into therapeutics, for example. But initially it was just to try and understand things. But then the other person that would have been in the room if we could have had them was Dave Anstey, right? That Jan already mentioned. And I think he had the bigger picture, which was he was the one who took the biggest risk because he put a lot of money into producing blood at large scale. And he was able to do that because he was part of the blood service and had that funding. And so the blood service and us went on that journey with him. And then we started producing blood at large scale in the lab from stem cells. And then from that onwards, we then did a clinical trial. And the motivator there was people with rare blood groups who can't currently have a donor because one doesn't exist. Or, for example, people with sickle cell disease or beta thalassemia, where they've made antibodies in their body against blood. And so you can't find blood for them because they've got these antibodies. And so you have to grow blood to be able to fill that problem, that clinical need.
So I think I had assumed in my naivety, I will admit, that this was in some way synthetically derived rather than derived from stem cells. So, and that's the tailorability of it, right?
They're normal. And I think this is the word we try to stay away from. They're not synthetic.
They're not artificial.
They're not artificial.
Which I think I had assumed they were, because I don't think in my head I'd made the leap to being able to create, what do we call them, natural, real, and at any kind of scale. But that is what you do.
Yeah, I mean, essentially what we're doing is we're taking, so your red blood cells are made in your bone marrow, and they're made from a type of stem cell. It's called a hematopoietic stem cell. Can be all your blood cells. Some of those stem cells come out of your bone marrow and circle it around your body and back to your bone marrow. And you can collect those from your circulation. So when people donate blood, they're part of the waste product. So we can collect those stem cells and then culture them in the lab and make red blood cells in the same way.
The idea is so, for example, if you have some rare donors, there might be 1, 2, or maybe 10 people in the UK that could provide blood. So imagine if you could grow more blood from the blood they provide, and then you've got more blood again on top of that. So I guess that was the original kind of nub of the idea, is can we produce more blood? And then some of the innovations that Jan and Dave had then led us to realise, well, actually we can make blood that's more universal to go into even more people. And then it kind of like grew from there.
I suppose what we should qualify here slightly is when we're talking about rare blood groups or rare blood group donors, most people know about the ABO blood group system, maybe the rhesus blood group system. That's 2 blood group systems. There's 47 blood group systems, 48 blood group systems now, with something like 350 different variants you can have between people. So when you're talking about a rare blood, we're not talking about
AB negative or something.
No. We're talking about a whole plethora of the differences 2 people can have between their red blood cells.
And in certain instances, we have this thing of like the universal donor and the universal recipient, but that's actually a simplification.
Yes,
there are certain
very
people
much
need
so.
very specific.
So it's true. So if you're in an accident and you got taken into a hospital and you needed blood because you had, like, say, a head injury or something, essentially, if they don't know what your blood type is, you'll get O negative blood. But that's the only stipulation, right? And if you need it there and then, that's what they give you. But there's a chance or a risk that there's not a matched blood group in there. And then if you have more blood at a later date, you've made antibodies against it. And then you can't have that. You can still have O negative, but you have to have a more matched product.
Yes, I see. Okay.
So for an initial thing, it's fine, unless you have a very rare blood type where there's maybe only a few people. So one rare blood type is called RhAg null. So it's a protein on the surface of red cell. So all blood groups are proteins or sugars that are on the surface of red cell. And these people lack it completely. And there's maybe 8 in the world.
Wow.
And they actually are more universal. If they were O negative and they have this RhAg missing, they're more universal for everybody else. But unfortunately, there's not many other people to give back to them. Do you see what I mean? So quite often,
I do, yes.
So for them, they would have their blood frozen, for example. There's a frozen blood bank in Liverpool. But in the end, hopefully you would grow more blood for them eventually when you can get to that scale.
And then the other one is people with red blood cell diseases like sickle cell disease. I think a lot of people have heard of, maybe beta thalassemia. But some of these patients, some cancer patients, have to have blood transfusions every 3 weeks. And therefore, they get blood from different donors. So it's going to be lots and lots of different mismatches. And they'll keep generating antibodies to the different donor blood until eventually you get to the situation you can't actually.
No, and these people are already sick. That's why they need so the last thing they need is this additional challenge.
But a sickle cell person would have 12 pints of blood if they're having an exchange, right? So the chances of a mismatch is quite high. Although now they're able, using new technology, to sequence donors so they can match really well. So that going forward, less people have blood that's not matched.
But it's still dependent on finding the matches among who's donated blood within a reasonable period of time, I'd imagine.
I think in sickle cell disease, about 30% of the people who have sickle cell disease will make an antibody against the blood that they've been transfused with. And quite often they'll have at least 2 or 3 antibodies, which then makes them impossible to transfuse. We can grow blood in the lab from stem cells, but there was 2 other pieces of innovation that, in the meantime, you have this new company that's in the States. We can talk about what that is later, but we have to have something that distinguishes us away from that, have an advantage. Otherwise, you're not going to beat this company that's super well funded.
Exactly the same stuff, then
but we realised that they have a few problems which we had the solutions for.
And they wanted our solutions, and they tried to talk. They talked to us many times and wined and dined us for our solutions.
But we doggedly didn't give anything away. So one of the key ones was what Jan and Dave developed.
So the issue was, it's all very wonderful. We can get those stem cells. We can put them in a lovely culture system in the lab to make the red blood cells. But these stem cells are so keen to mature or turn into red blood cells. You can only make a finite number of red blood cells from every collection of stem cells. The cells will divide sort of a certain number of times on the way through, but then they're going through this dynamic changing process. They're spitting out the nucleus to become the red blood cell at the end. So then you've got to go back and get more stem cells. And if you do enough from a rare donor, that's really limited. So what we did, we took those stem cells, we pushed them, matured them to the very early immature red blood cell stage, and then we immortalise them.
Okay.
And so they keep going forever. And then when you want to, you can take away the agent that keeps them immortalised, and they trundle down and become the red blood cells.
But you've got control over that rather than
We've got complete control over this, yeah. And then the other cool thing is CRISPR came along, that lovely gene editing technique, which just opened up a whole range of sort of possibilities for us. So now we can CRISPR edit those cells.
Yeah, so for years there was ways of gene editing cells, but they were all either impossible to work out, which was talons, and then there was another one called zinc fingers, and again that wasn't that accessible. And then CRISPR came along, and I was like, I understand this. So I was like saying to Jan, we got to do this, we got to try and remove some blood groups. And eventually then also Jan's lab started making cell lines and things from them as well, with for sickle cell disease and things like that, to try and study the process. But sometimes it's like having a technology that then suddenly opens up tons of doors into new areas and new possibilities.
So it did. So basically, because what you can do then, so you just have the immortalised, we call them erythroid cells for the immature red blood cells, just banked in, frozen down and kept. You can thaw them out when you need them, and then we can CRISPR edit them. So yes, we removed 5 of those problematic blood groups, a proof of principle of a much more universal blood project. But one thing my lab is doing now, as Ash mentioned very much, is studying red blood cell diseases, and there are a lot of red blood cell diseases. It's very difficult. You can't get the material from patients, they're anemic, so they're hard to study. But now what we can do, we can introduce those disease-causing mutations into our erythroid cell line, which is we can create the disease in the lab, and then you can study the disease and you can screen drugs.
Brilliant.
So that's where my lab's gone. That's an angle. It's not quite Scarlet.
No, but it's complementary, I guess, isn't it? Yeah.
And then the other thing that was kind of the other extra thing was that moment when we held blood in our hand, and like literally in our hands, in a tube. And then later when we started, just before the clinical trial, we were doing larger volumes, and you could see this large, it's a mini dose, right? Enough for a baby, not for an adult, but still a significant, like billions and billions of red cells. So I guess my eureka moment was, we need to put things inside the red cells because they're designed to travel around the body. And that's what I tried to sell to the university originally when I wanted to set up a company. And that's what the new company in the States kind of did. What I realized was they hadn't, after talking to this company, they hadn't really thought of all the problems, but we had solutions because we'd studied disease and understood the process. And so that's where we started then expressing proteins inside red cells to make a red cell with something inside, which is a therapeutic red cell.
Okay, because my question was going to be to what ends? Because having disclosed I nearly studied biochemistry, I definitely didn't.
So imagine the simplest is you imagine you've got a disease. Which is missing an enzyme, right? And so the simplest thing is what Scarlet's going for at the moment. There's one called hyperammonemia, which is essentially really, really high levels of ammonia, right? So that's toxic to the brain, causes lots of problems. People are really, really ill with it. You get it with liver disease, for example, and also some urea cycle disorders. And so what we can do is, and what Scarlet's working on, is producing a red cell with enzymes inside, which can metabolize away that ammonia. And then you don't have the problem anymore.
So the blood is the medicine in extreme
literally, that's literally our strapline, I think, for Scarlet, is we produce a new type of medicine using blood as a vehicle, and we hide the medicine inside the blood cells. It's not recognised by the immune system because it's hidden inside.
And because it is compatible with that individual's blood, so the immune system just sees it as more blood. And if you've got cell lines and a more universal cell type, you can give it to more people. So it's not the case of you are a patient with a blood disease and we will use your stem cells to create blood for you. It's more that the blood you're creating is compatible with the right wide range of blood types and individuals. Okay. Brilliant. Having put those bits of the puzzle together, the point at which the university says, actually, yes, we will support you to make this company. What did you do? How did it go? Did you seek investment funding? What was that initial stage like? I remember. So we were thinking about it, and then at the time you're thinking, how do you go about this? So initially I started talking to Harry Dequire and Science Create, but then started talking to some VCs, and then actually got approached by a VC when we published some of our papers. And then Jan and I, plus the other people in the team in our labs, certain ones came in who were interested in doing a spin-out company. And then we worked with this VC with the idea that it was going to be quite a lot of money. So this is the hard bit. So we worked for 9 months on a venture build with them, but we kind of didn't agree on certain things, and then it fell apart. And then actually at the same time, in parallel,
one was going on, and that got funded, right? So we saw that one get funded, and ours didn't.
Frustrating.
And so at that point, you question, should I bother going any further, right?
I feel like I've tried this twice now,
Yeah, but then actually I was like, nope, right, this time we're going to just do just a couple of us, and we're going to just really push through this. So we did two things. One was we started talking to Science Creates again and some of the other VCs. And then separately, we approached an accelerator in Cambridge. And this was the idea of the university commercial team. They were like, try an accelerator, see if you can get, because often with accelerators, at the end of an accelerator, you have VCs and investors that come in to listen to some of the ideas. Sometimes you might fund something. And so we got accepted onto the accelerator, which was really exciting. And we were going to join the boot camp. We almost signed the term sheet because there's some equity you have to give with some accelerators. And then separately, then Science Creates said, we will do a deal with you. Sign this term sheet. And then we also got another investor, which is the Abcam millionaire investor, Jonathan Miller. And then that's how it all happened.
And I guess with the accelerator, it did transpire we were already a bit too advanced. They were a bit surprised, actually. Normally they have people who have ideas, but haven't actually put them into place. So I think we're a bit unusual.
We've gone through that process with the first VC of coming up with some ideas of what we would treat and also like programs. And so we got quite advanced.
And we had the technologies as well, though, rather than just an idea. We actually had them physically.
But if it simply allowed you to confirm your initial thinking and be even more confident in what you wanted to do, then I guess it still has some utility.
And it's exposure to the actual field sort of thing, or into the area of entrepreneurship. Yeah, I mean, they were great.
I think one of the key things you have to remember when you do this stuff is it's all a journey, right? So actually one of the reasons why we didn't progress with the original VC was because we had a disagreement around certain things. And then as you go on, you realize that what they were saying was right. It's not always the truth, that sometimes you're right. But in this case, we accepted that they were right, but it was too late, right? But the door was still open, and maybe one day they will invest with us. But sometimes you have to come to your own, you have to accept it, because it was too early to accept it, because we didn't want to put everything into one basket, if you see what I mean.
I completely get that. And I do think as a founder, as an academic founder, there's this real balance between humility. Sometimes someone else, in this case an investor, actually might know what's the best route, and maybe I don't because I haven't done this before and it's new to me. There's other times when, and I can think of examples in my own journey, where I had a set to with somebody, might have been an investor, might have been a colleague, whatever, and I look back and go, no, I was right. Like, I stuck to my guns, even though I felt out of my depth, and I was right. So there is a balance between the humility of knowing that you don't know much about this world, but actually the confidence to nevertheless, and say, well, this is my innovation and I am going to drive it. So that's
also you just need to get more evidence, right? That's the other thing. So if the timing is not quite right, you don't have all the evidence. Sometimes it's hard to make that leap. And I guess going backwards, I wish I had a long time ago just backed myself. When I reflect, I kind of realised that I should have just gone for it. But then I would have maybe had a different career and all sorts of things. I think you have to come, when you come to spin out, I don't know if you thought this, Jan, but like you kind of have to feel comfortable in your skin. This is what you want to do now. And you can't write more. Well, actually, Jan still writes millions of grads. But I find I want to do something well, and I really want to take this to the clinic. So it's a real focus of mine. And so I can't apply for all the grants that I want to. So the lab suffers. But the way I think of it is, well, I'll do this for now, and then I'll change it later.
Figure out the next bit when I'm ready. Yeah.
And actually, you just go on a different journey. So, and it's amazing that as an academic, you can choose that journey, right? You're in charge of it. Okay, you've got Nigel will be listening to this now, right? Our head of school, he'll be like, you need grants and everything else, and overheads and everything else. That's completely right, and I completely agree with that. But also, I think it's also about yourself and what you personally feel comfortable with, what you're excited by. Because if it was all grants, I think I would just do a different job.
I think also, though, that exposing yourself to the different world also gives you ideas for grants. Because you're exposed to different ideas, different ways of thinking, and most of our funding nowadays really has to have some sort of ultimate purpose.
Some pathway to commercialisation or impact.
So it kind of exposes you to that and gives you a different way of thinking, and you'll probably come up with a grant you wouldn't have come up with beforehand.
That's a really interesting you bring that up, because it's quite a recurring theme in these interviews and other people I speak with, is you go down a commercial route in whatever that means to you in your field, but there's quite often a sort of feedback loop back into your research or into your colleagues' research if you've actually left the academic environment. So it can be quite almost like a virtuous circle. It can feed back into the research.
Because a company can't do everything, but you can take an IP at foot, and if the company then wants to license it, that's good as well.
But sometimes the frustration with companies is something happens and you know there's something academically interesting there, but the company won't do it because they only have so much money to do what they said they'd deliver,
And focus,
right?
focus.
And so you can only pick so many of those ideas up as well, right? So that's the other thing is something, oh, this is really cool. And if as an academic, it's like you got to be really careful you don't ultra focus on the thing that's academically interested and lose sight of what the business is, right? So that's something that we've learned as well.
You use the word learn a lot and you use it with glee, all the things you've learned. I think there's also a courage required of academics, let's call us career academics, experienced academics. We're none of us straight out of our PhDs, right? There's a courage to move into a space, entrepreneurship in this example, where you know you've got a huge amount to learn, where you know you're not an expert. We trade on our expertise. Those are our laurels. Those are what we're known by, is what we know a huge amount about. And to take a step into a commercialization pathway, unless you're doing it for the second or third time, chances are you don't know a lot about it. So you seem to talk about it with glee, but has that been hard? Have you struggled with that? I'm not an expert in this and I'm going to show it?
Oh no, I'm not bothered about that. But I think we did have support. I mean, we did have was it, can't remember the name?
Yeah, Laura Newell.
Yeah, I mean,
so initially we had somebody in the commercial team, commercialization team, to hold our hands a little bit, and that was really helpful. And now we have other people who help us with new developments.
Andrew Wilson.
But actually the job itself, academia itself, is a sense you pick an area you don't know much about, right? And then you go and explore it. So it's just a different type of...
It's just as scary going to conferences in America, you know.
There's elements you don't like, and I don't like being wrong, right? That's kind of like, if you're in a company or if you're an academic, you never want to be wrong. But actually what my mindset is, sometimes I am wrong, I accept that. But actually when you learn out you're wrong, you've learned something, right? So this is probably why we like learning this, because you will make mistakes. If you run a company, it'd be amazing if you did everything the way you planned,
and almost impossible as well.
Yeah, but hindsight's a great thing though, isn't it? Because you don't know until you've tried something.
No, absolutely.
But actually what's cool now is you can bring stuff in from what you've learned from going down the commercial route and take it entrepreneurship, and then you can bring it back to the students. So quite often now I'm asked to give talks to explain it and try and infuse them and see if they want to go on this journey. So and that's kind of interesting. It's really interesting because everybody likes talking about themselves, right? It's easy.
Absolutely.
And so you just have to stand up in front of students and talk about a journey and what you've done in your life.
And let them see your enthusiasm. But I mean, we do a lot with students to try and expose them to different career paths and options and so on. But the difference is you're their academic, you're their lecturer. They might have seen you giving the year one tutorials, whatever.
They see both sides.
So they see someone that they already respect in their discipline as having a different dimension. And that I think can be, well, it's a different kind of powerful than bringing in someone who is just a commercial person, It's
more accessible to them. You can see accessible route. I think the good thing about our field as well is red blood cells, people understand what they are.
Yes, that is also good. It's a good bus stop conversation starter. And
everybody's cut themselves and saw some blood. Like people might know somebody who's had a transfusion or had an issue.
They understand it, which makes it much easier to talk about to people and to students.
And that's why the media, quite often the media gets really interested in this stuff. So, as long as you've got something interesting to say, quite often it gets picked up by the media.
For sure.
And there's a lot of people around the world because blood is so accessible. There's a lot of labs trying to do various things. There's not just what we're trying to do, which is growing blood, but there's also creating artificial blood, true artificial blood, which is essentially proteins that are in blood cells surrounded by an artificial liposome or some other cage, and trying to make that work inside the body.
And what's your position on that? Because that's not the approach you've taken. Do you think it has utility? Do you think
it is already used
in an emergency?
Okay,
absolute emergency, but it's not going to last very long.
It doesn't last. Literally a few weeks ago, another announcement from another group, this time in Japan, that they had purple blood.
Interesting.
So they had they altered. So hemoglobin is what's blood is, it's the red of blood, right? So it's the thing that carries oxygen, and they'd converted hemoglobin. They've bound something to the outside of hemoglobin, and then we're using that as an artificial blood type. But the thing they bound caused the hemoglobin to go purple.
So it's still hemoglobin. They haven't.
Apparently, I don't know enough about it, but interestingly, it might work. So we just have to wait and see what happens with it. So But, people have to try stuff, I guess. And then the thing that works the best will be selected.
At the end of the day, though, you kind of think, our bodies are pretty spectacular, and creating something completely artificial that works as well is going to be a massive challenge. Yeah. Whereas actually making red blood cells in the lab, they're the same as our normal red blood cells, and they last 120 days or whatever.
So it's kind of like a no-brainer. If you can make enough
it's
red
a
blood
natural.
cells,
It's like natural medicine.
it should last. If it's identical, it should last as long as normal blood cells last, which is 120 days. Right?
And do you know how long it lasts yet? How long
we're literally doing the Restore trial now, which is a clinical assessment of lab-grown blood versus the same. So lab-grown blood grown from stem cells. So from a donor going into a participant or volunteer who is not related to that donor. So like a normal donation, essentially going into somebody who needs the blood. In this case, a volunteer, they didn't need it, but they're doing it to help science. And then we take the same donor's own red cells, and then they're being compared to the lab-grown blood in terms of how long they circulate the body. I've been saying this for several years now. We will know soon what the result is, but because we set out and it was completely blinded, we even though we've nearly finished, we still don't know what the result is.
No idea what the actual
So my guess is they're going to be equivalent, because there were some animal studies that I have done, but other people have done, and that suggests they are equivalent or maybe even possibly better.
We're hoping they last a bit longer because they're all new.
Yeah,
I guess so.
When you get a bag of blood, a lot of them those red blood cells are destroyed immediately because they're
some
aged.
Some old ones, some young ones, some middle-aged ones.
Yeah, precisely. But these ones should all be nice and new and fresh. So hoping they're going to last longer. Yeah.
So is that the main priority for Scarlet at the moment? So what's Scarlet's sort of main to-dos at the minute?
So the thing is, at the moment, I guess one of the things we had to accept was we're not going to be just making unmodified blood, because at the moment the money is not there to. I could take blood at your arm and it goes into the NHS via the blood service, and the NHS pays £130. That's not enough for an expensive self.
No, I see. It can't just top up supplies because it's too easily available from normal humans.
The driver for me is to produce blood that can go into anybody. But at the moment, it's a therapeutic blood where we can generate enough money and then get economies of scale and other things, which then will allow us to then take unmodified blood into the clinic
and produce it much more cheaply because you're doing it at scale.
So the more expensive medicine blood becomes the driver for then unmodified blood to make that effect.
Are there particular targets then for the medicine blood?
Yeah, so we've picked indications. So you do a study, so that's on the Scarlet website, so people can look at that if they want to. But there's a whole list of things you could potentially do, lots and lots of metabolic diseases.
But that's, from my experience at least, in the spin-out, that's part of the difficulty. We're researchers who want to investigate all kinds of different things, but you've got to focus.
You have a program, a product. I'm not going to say what the products are exactly, but the program is pushing forward a whole bunch. It's like a competition, right? Okay. We've got an internal competition to get the first one to the point where it could potentially go into somebody, right? So they're all competing. There's not a massive team. There's about 7 people in Scarlet now, right? But essentially they have multiple products. Okay. And then one of those products is going to be the first one that will hopefully go into a clinical trial.
Some of them are super exciting, though.
That's another thing that you're working on at the minute. And
And the super cool thing is by being in the company, it's ultra focused on that thing, not trying to generate a paper, not trying to get a grant. Well, actually, if we got a grant, great. And Scarlet has got grants, Scarlet has got Innovate grants. But the thing is that tunnel vision of we have to get for Scarlet to survive and be investable, it has to have a product which could be taken forward. So it has to be a competition internally, and the best one with the most activity is the one that's going to go forward.
That will go forward first.
But I can't tell you where it is.
But also an area where there was a clinical need.
Yeah, for sure. I'm assuming all of your candidates
so everything
had a clinical need, but
where there isn't any treatment at all, or the treatment is
inadequate, or
So everything will be weighed up. Then the final results will then lead to that clinical candidate.
I'll need to interview you again then, because I want to know.
It moves quite faster, doesn't it? We've got a great CEO, actually, who's good at driving us.
That's a great area to explore, because I'm always interested in the decisions academics have to make when they decide to having decided to found a company, so that decision's made, there's then a myriad of small decisions need to be made, and including what role you take. Do you leave your academic job and become the C something, the COO, the CEO, CX, whatever? Do you do both? Do you actually just spend half a day a month attending discussion, strategy meetings and otherwise stay in your academic. Whatever decision you make, there will always be other skills that you need. You'll always need somebody to do something that you just can't or are not interested in learning about. And I think identifying a CEO with the right skills and the right alignment with you as academics is one of the most difficult. There are great people out there, but that alignment's got to be great. Well, how did you? Well, yes, lucky, but
you go with a gut feel, right? But I don't know for Jan what she felt, but I knew I didn't want to be a CEO, right?
because
I would have liked to have been one, but I didn't want to give up my academic job.
okay, so it was more that you weren't,
and it wasn't because I didn't
want
believe in the idea. I just thought, I don't think I'm the right person. I can lead a team, that's no problem, and I can do that in the company, it's no problem either. I think what you need is somebody who's good at business, and academics are not necessarily good at business, right? So if you've got a business head, great. Obviously, you can come up with, there's indications to target and things like that, but really what you want is somebody who's experienced in business. And I think the quicker you come to that decision, the better.
But coming to that decision, then finding the right person.
Yeah, so we had a competition.
Yeah. You had a competition. You like a competition, don't you?
Yeah. So I love a competition. So it was partly around. So nobody would explain to me what was in a, when you write your business plan, right? So it was kind of annoying because they're going to rip up the business plan anyway within however much time. And everybody's constantly going on about business plans. And I was like, what does one look like? Now you can just go on ChatGPT and just write a business plan really quick. Yeah. But then remember, you're giving your ideas away. So you have to be really careful. Just ask it to do bananas or something like that. But that's what I do. But anyway, I just did it for a test to see if it's possible. I wrote one in 30 seconds, right? So they kept going on about it. And nobody could really explain to me what's supposed to be in a business plan. And there's nothing online really other than some things saying you need to put a few things in a business plan, but it's no. As an academic, you need an example because you do for a grant. You want somebody who's been successful, so you can compare
what questions
it.
Do I need to answer?
But nobody gives you a business plan, so I was like, right. Well, so then you definitely need somebody to help you do a business plan. So actually, what we did in the end was, and then this was with the commercialization team, is we found some people who potentially could be CEOs. We had a friendly enterprise entrepreneur who suggested some names who had just been filtered for another company. And Alistair Irvine, who's our CEO now, was one of those names.
Okay.
And we worked with them to come up with a business plan and also work out where did Scarlet want to be and all this other stuff. And in that process, we did our selection.
Okay.
It's who
so
can you work
each
with?
of them, and then sort of almost found which one seemed.
So we started with 3. One got jettisoned quite quick, and not for personal reasons or anything, just what happened. And then the other 2 we worked with, and then we basically went with Alistair. So
it didn't seem a hard choice.
No, and it's kind of what you're looking for is, I think, somebody who's as enthusiastic as you about doing it. And that was part of the test as well. So in my mind, I was looking for somebody who wanted to make this successful and take it to the clinic, who had a brain that was business, and also was a negotiator because there was some IP that we needed to negotiate. So we were looking for that. And I kind of had this tick box in my, like a list with tick boxes in my head, and Alistair ticked all of those. And I'm sure that was the same for Jan. I don't think we even discussed it. We just said
we discussed ones we didn't like or didn't want, but
in the end,
But also, Alistair has, luckily for us, a scientific background as well.
Okay.
So he's involved in discussing the research as well.
Okay.
and he's really good at saying no.
Oh yes.
The enthusiasm and enough scientific background to be able to have proper scientific conversations, but the differentiation from you and him, or the CEO in general, will be the more commercial mindsets, more negotiation.
And that's kind of developed, hasn't it, as well?
Yeah.
I mean, it's shifted.
What about values? So sometimes when I've observed conflict between the academic founders and other people they bring in, is if they're equally enthusiastic to get that innovation to the market, whatever that is, but if they have different underlying reasons for wanting that, or different systems of values that inform that, I've seen that go quite spectacularly wrong.
I think that's where you've got to accept that in the end your baby might get sold or floated on the stock market or wherever, right? So, or actually it might be bought up by somebody else and taken apart. And I think you have to come to that quite quickly. If not, you need to run the company yourself, right? But then will you ever make a success of it? I don't know.
I think that'd be difficult. But even if we lost Alistair, we had a different CEO and it could change. It'd be like,
Yeah.
Don't want that particularly either.
There's compromises that you make as you go along because you realise that it has to be a business, right? So, and it can't be academic, and it's continual that you make these compromises. But you come to an agreement and an understanding of those people. And I have a respect for him as a business person.
Yeah, I think that
right?
as
So,
well.
like,
It's
I
also
trust his
respecting that he
decisions.
does know a lot that we don't know.
But I know that he will work for the good of the company rather than the good of me. Right.
Yeah,
That's the
which
other
is,
thing,
well,
which
that's
is what you
fiduciary
want.
responsibilities right there, but it
Yeah.
is also, trust me when I say some people find that harder to get their heads around than
Yeah, no,
others.
I-
you
I-
guys have, and that's great,
I
but it's not
realised
universal.
it took a bit of time.
Yeah.
And also when I think the thing that I found a little bit crushing at the start was when I had a target group of like a disease that I wanted to do first, but it wasn't big enough.
Yeah.
And there's no money in it.
Yeah.
And then there's the issue that actually, and I never knew this, but like if you start out with that really small group of people, you've got to be able to give them that drug, but then they've been treated by other people. So like the chances of finding somebody who can take your drug is minimal.
Yeah.
But then if they do take you have to give them that drug for as long as like it becomes available until it becomes widely available, right? But then if it's not a very big market, it's never going to pay for itself, which is really crushing for that disease.
Yeah.
When I realised that, I was just like crushed about it. But then that's what made me think about other options.
Other ways to get this underway and then address some of the more tricky ones.
But the pattern started with that particular one. That was the start of the journey. It had a role.
So tell me a bit more about your respective roles with Scarlet now. So Ash, you didn't want to be the CEO, but you were the one really pushing the formation of a company. Jan, you were the person he talked into coming along on the journey. So maybe we'll start with you. Like, what role do you play and what do you get from it? What do you do?
Got a consultancy agreement with them.
And what does that amount to in real terms? Do you sort of provide like scientific advice?
Yeah, at the moment, we have a management meeting once a week. We have an R&D meeting we go to once a week. We feed into the science. We go through the data. So very much, it's not so unlike being in my academic lab.
Well,
really.
I think it sounds like a research group meeting. So is it like that, but then there are some different boundary conditions because this is a company, not a research group? Or does it actually feel quite similar?
At the moment, the R&D meetings seem quite similar sort of thing. It is really sort of, the biggest thing is the conflict of interest between the two and keeping your research, because we are in the same area here, but wanting to feed into the company. And then I suppose the other meetings, more on the management side, are the ones that are more alien. Yeah, really. There's a lot of fundraising sort of thing. Really.
Do you get involved with that fundraising?
Alistair is also hugely leading on this, fortunately. It's fun at the moment. It's good fun, and I suppose that's what drives it. And we know, I mean, two postdocs from my lab have joined, two postdocs from Ash's lab have joined, and we've had new people. So we know the people down there. It's sort of a bit of an extension of what we were before, really. And as it's close by in Bristol, it's not like it's far away to go there, really.
Over time, it becomes like you're more advising. So initially you're driving because there's nobody, right? So you're starting from nothing. You've got to pick the people, but if you already had some of them in your lab, then great. If you know this,
you know what you've got. You've done a 3-year interview.
And they have the experience doing what we needed.
And so you can trust them to do stuff, and if they say they're going to do it, they'll do it.
And you know the things that they haven't got experience of, so where you'll need to allow extra time for upskilling and so on.
One of the key things you have, like Jan mentioned, the conflict of interest, you have to really, really be aware of it. And it's so easy to you could just keep handing your company stuff. So at some point, some elements of knowledge transfer occurs, but then you have to wear hats. Yeah. Right. So I'm like, which hat am I wearing today? And and you have to be challenged by your head of school and other people. Are you thinking from your company side of things, or are you thinking from a academic side of things?
And conversation. Is yours a similar arrangement? Do you act as a sort of
so I'm a consultant. So we're both directors for the company. We then are part of the management side of things. And I imagine over time we'll become less and less involved in that side. We'll almost certainly be involved in advising the science. It's kind of weird because when I first set it out, I didn't know how long the relationship would occur. I wanted it, like, you want to be involved all the time, right? But realistically you can't, because you've got a job here and you want to get back into that and do other exciting things as well, right? So kind of getting a balance between the two things. You do a lot of outside work, like so outside the normal academic job, which has no hours when you think about it. It's a challenge, isn't it? Nobody ever tells you that. It's not a 35-hour week. It's way beyond that sometimes, right? Especially for you. Chan will work many hours.
I can't imagine letting go of the company or not being involved.
Because it is like a little baby.
I'm not even going to think about that, actually.
So one of the things I said earlier on I was keen to explore, and we started it, is the fact that you're academic co-founders. Your research seems to be quite in adjacent areas, quite closely aligned. You both have bags of enthusiasm, bags of thoughts on this. What is your relationship? Do you always agree? Do you challenge each other? Do you take each other out in discussion? Do you sort of...
Well, we're both wrong sometimes.
Do you joust in these research meetings in the company? Like, how does that work?
I think we bounce off each other, but we definitely disagree. Ash has the most mad ideas you've ever come across a lot of the time. He has really blue science sort of ideas. But also, it's wonderful because I can go with something I'm not sure about and talk to Ash about it.
So,
it's
sort of be a counterweight a little bit when he's got a mad idea that you think is too mad for mad, but...
Yeah, but my back is completely, got my back completely. Yeah. So it's always like, I know whatever it is, I can phone Ash up, talk about it. If I'm worried about something, I can go and talk to him. If I don't understand something, I can go and talk to him.
Yeah, but we do
so it's having
goes
that
both
nice...
ways. It's kind of like... And Jan is a real people person as well, right? And I lean on that quite a lot because I'm just excited about lots of things. So there's kind of like sometimes you need somebody... Like, you're a realist as well. I think that's really like maybe more... touching the ground.
No, definitely realist compared to you,
I'd rather not be confined, so I will try and do things that some people might think are impossible, but I'm like a half glass full rather than a...
Yeah, but that's where the ideas then come from, don't they?
Yeah, but then sometimes it doesn't work and I'm wrong, right? But then you've learned something as you go along. I think that's part of the excitement.
And you both have that willingness, it seems to me, to put those ideas out there to each other, which can be exposing. If you've got an idea and you think, I don't know if this is going to work.
Yeah, it doesn't feel like that though. No. I mean, maybe like somebody else it would. I could feel inadequate if I came up with a mad idea.
All ideas are good ideas.
Yeah, no, that it does. Yeah, we don't have to feel. So I wouldn't. Yeah. So yes, maybe that is a different relationship to other colleagues.
I should say as well, because there are some researcher co-founders, so in Scarlet as well, and I guess they're more realist. So they quite, at least one of them is. And so quite often used to counterbalance my mad ideas.
It's quite grounding, But they're great as well. So I think we've all worked together for a while. So we're all really good friends.
And it makes it fun,
which is nice,
You mentioned, if I understood you right, Jan, you do your university research group meetings jointly as well. Is that what you said? That strikes me, it's quite an important thing to model to your PGRs and your postdocs, because if you're able to challenge each other or pull each other down or push each other up in terms of like, that's a silly idea, that's a brilliant idea, whatever it might be, you're modeling that kind of collaborative, trusting, be able to say what you like and not be worried if somebody thinks it's silly. You're showing that, you're living that, rather than saying to them, you should do this, you're literally doing it in front of them every week. And that, I think, is quite powerful. That's not the sort of thing that would have happened when we were PGRs, I don't think.
It's kind of respecting what somebody else's idea. I think hopefully most academics would respect somebody's coming in. Like, they might be wrong, but then you kind of could just say, let's think about that a bit more. You could do it in a way that's not disruptive. So there's some mean academics around, right? I've seen some.
Well, there's mean of every profession, aren't there? But I suppose what I'm saying is, I think we'd all agree with the principle, but modelling it week in, week out in front of your research group is something that not as many people would do. They might say, this is what we should do, but actually literally doing it live is quite powerful.
Yeah, but it's all quite happy and jolly. So.
I guess it's not a conscious thing. It's kind of just actually, if you do this job on your own, it could be kind of lonely, right? Even though you've got people working around you and they're doing your favourite thing, and there's nothing better than somebody coming in and going, look, I've got this, and nobody's ever seen that ever, right? But then actually, if you can share it with somebody and then also come up with new ideas together and go off in new directions that maybe you never thought of, because there's two of you, then it opens up a vista, right? So,
and I think I shouldn't lead the witness, but I think this is kind of what I'm trying to drive at, is from a sort of, let's call it a research culture point of view. The traditional model is more of the group lead and their people, and then down the corridor there's another group lead and their people. And I know that's not always how it happens, but going back, that was certainly the more common model. By modelling that partnership, discussing, having ideas, just being really open, you're setting that as the norm for the next generation of researchers that are coming through. Jan's idea might be something that one of Ash's students works on, or vice versa. That openness, that sharing, are not competitive with one another, but rather collaborative.
It also increases our productivity.
Yeah,
and that exactly does happen.
You get instant feedback, right? So like, whereas you might have worked on something for ages, when actually if you just get the idea out there. But actually in the building, we're not next to each other. Like, it would make sense for us both to be in the same
different
lab,
floors.
but we're actually on different floors.
So it'll often be somebody in the lab or Dash's lab or thing, really. But it's great for them because they've got an extended team of people, essentially. It's not just one's in my lab, one's in Ash's lab. They kind of know if they want something, they can, if we run out of something,
it's a pool of expertise.
run on down. But it's the norm to them as well, though.
And that's exactly what I'm saying, because they will then perpetuate that if they have their own research groups going forward, I would hope,
Well, otherwise it's kind of lonely, like I said, I think. Because you're just that
You're kind of
person.
isolating, can't you?
For sure. Funding a small and growing company is, I think, challenging at the best of times, but you're a wet lab company, you're a biologics company, I guess. So that's one of the hardest challenges because it costs a lot of money. How have you funded the company to date, and how have you found it?
So when we set out, we had that first instance where we were potentially going to get a lot of money. And that's the gutting thing, right? When you go from a lot of money on the table to less money. But actually what happened was when you go through that process, you realize what you can do with kind of a certain amount of money, how much progress you can make. Essentially, you have to get to an inflection point, right? That's what every investor wants you to hear. And as you go along, this is why the business plan sometimes gets ripped up a little bit. But actually, to be honest, ours hasn't yet so far. Probably because I didn't write it, but I inputted into it, but I didn't write it. But essentially, you have to kind of think, what can you get the minimum done, but gets you to the closest to an inflection point? And then that's what the investors buy into. And so I worked out that we needed about £2 million. Probably get away with 1.5. But, if somebody had given me £250,000, I would have still set up the company. But you wouldn't have managed you can't do any therapeutics with £250,000.
Absolutely,
you could just start. So you have to be realistic. So I thought 2 million would give us probably a runway of 1.5 to 2 years. And we also had a little bit of income around the cell lines as well. So there was a little bit of cash coming in. So initially we got investment from Science Creates Ventures. And then also Jonathan Milner's vehicle, which is called Meltwind. And then I guess the whole process, what I didn't realize is you're instantly now thinking, where's your next lot of money coming from? Which I didn't realize. It was like, I've got money. It's like when you get a grant at academia, you're like, yay, I've got, especially if it's a 5-year grant, you can like just go, go on.
So yes, we got the money, and initially we didn't have a premises. So we were sort of doing the work in our labs, and that is really expensive. Even though you managed to agree not to go in more than 100% sort of overheads, so much of the money was actually just disappearing. And by moving out to actually...
That makes things go faster. Actually, become more credible when you move out, which you don't realise.
Yeah, I think so. And then the provision of like lab space for small but growing companies in Bristol is so different now than it was 5 and 10 years ago. It's really
that was another bit for me when I was thinking about it. Is where do I go? Where do you go? Actually, now with what Science Creates and other like Set Squared and Engine Shed, there's lots of places.
But the fact we can stay in Bristol as well.
Which is practical for you, but also from my point of view, I want to keep our small companies in Bristol. They're employing our graduates and collaborating with our researchers.
But it's also close to where your labs are, right? So some other developments might then feed in, assuming there's no conflict of interest or anything. But essentially realised that we would save a lot of money just going out into Science Creates incubators. But there are other places you could go. But for us, it was perfect location. But they were building it at the time. And then we had to move in. We bought all the equipment, and that was still way cheaper than working in the university, which is you never really think about. So that was a real moment it all crystallised. And then, like I was saying, suddenly you realise you've got to keep raising. You need to have conversation. Initially, it's conversation with VCs to gauge interest, and then it's kind of like you're up against a situation where actually a lot of VCs don't have any money. Which has been a bit of a shock.
They can be as interested as they like.
So they're like, we haven't got the money at the moment, or the timing's wrong, or the tax timing is wrong, factors, which I never realised. So as we've gone along, I've realised how complicated it is. But you collect a group of interested VCs, and then you essentially you decide on the next part of your business plan, what inflection points you're going to, and then it's like, how much do you need to raise? And you kind of scale it depending on the interest and what's potentially possible. So you have a minimum, and currently we're funded with like 2 million. We had a bit of an investment with Innovate grants and a joint investment with an Innovate grant with an investor. And then going forward, now we're looking at 3 to 5 million investment, so the next stage. And then beyond that, it's probably I think we're looking for like 20 million or something like that. So when you're at the Series A, it's at 20 to 30 million.
It does feel like a bit of a treadmill sometimes, doesn't it? Always looking at the next tranche of investment.
And it's like shocking when you talk to investors and they can say something to your face and then you find out like you think they're really interested. And it's crushing later on that actually that's the face.
I always say an honest and preferably reasoned no is so much better than a polite maybe, which they don't actually mean.
Like any investor who ever listens to this, just be really honest straight away. Don't waste people's time, but also give feedback.
Yeah, absolutely.
Because if you're a good investor, if you give feedback, you can adjust.
Exactly. Or you can target a different type of investor. If it's because, and it's a reason that is not surmountable for that type of investor.
So my favourite thing is I hate a no. Obviously, everybody hates no's. But if you get an investor who's a really good investor, they'll say no, but I know this group are interested in this and they have some funding. And they should introduce you to somebody. If they're a good investor, that's what they should do. They should facilitate, right? So that's all this stuff is kind of fascinating because it's not what we see on the academic side because you just apply for grants. There's a process in grants, right? Reviewers and panels and everything else, but this is completely different.
Actually, I'd argue the academic process is quite a lot more transparent. They say that this would be the process and this would be the process. You can look up who's on the panel, so it's easier to navigate.
And also, a lot of the investors come across don't know anything about science. So you're trying to present something to people who don't really, are not in that area. And one thing we did learn is when you're sort of sending something to an investor, make sure it's really short and really brief because they've got so many coming in, they're not going to turn a page over.
They've got hundreds. Hundreds in a week.
He needs to really grasp it. But that's a really nice segue, though, because there's an importance here about the clarity and simplicity of your message. And you mentioned earlier, it's nice to work on blood and blood cells because literally everybody knows what they are. Everyone's got them.
Yeah.
As the company or just as yourselves been involved in quite a lot of public events, you've been interviewed in the press, you've spoken to a general audience, not an academic audience. Is that something that you kind of set out to do with an agenda? I want to do this to help my company succeed, to help my research, or is that something you just enjoy doing it and do it for the fun of it?
I particularly for either of those two reasons initially. It was just to share with people because you're excited about it. You want to share it with other people actually. And as I said, because it's blood, it's not a difficult thing for people to understand at different levels really. So I think it's just the fun of it as much as anything else.
And to tell people how cool it is. Not the company side of things, just the idea of growing blood and potentially altering it and manipulating it.
And the fun of research.
And actually when synthetic biology came out, because that's where a lot of the research that went into Scarlet came from, is the investment the government made in synthetic biology, and which is now called engineering biology. There was a worry that the public wouldn't trust it. So actually we did active engagement to try and explain what we're trying to do and the reasons. And I think actually when you're talking about therapeutics, I think people just get it. It's for the common good. And I remember asking like a group of public people, like random ages, random people, and I was like, what do you think? They were like, we trust you to do the right thing, which actually was really
fascinating. Wow, that's warm.
So it's not the evil scientist in the corner. It was more that they trust that you have ethics and values, which mean that you're trying to do the right thing for people.
We do have, as part of the BTIU, there was a patient, people attending meetings. They've been great. I mean, so they come along because they've got the disease that maybe you're trying to make the blood for, and they're really interested and really keen. So in some ways, explaining it to them and getting them to understand has been fantastic because you're getting it over
and
to
what
people.
Questions they ask.
Yes.
You can then hone how you explain it to try and answer those questions up front.
Yeah, absolutely.
So that's a public advisory group that we have that's linked to part of the unit that we have, which is called the BTIU. But one of the hard things is because it sounds so cool, but everything takes time, right? So it's also trying. So the worry with the media and interaction with media, especially, is that you say we can grow blood, but obviously there's a load of processes to go through before we can actually go into people. And but they want to just say we don't need blood from people anymore, right? But that's not true. Like absolutely go and donate as much as you can because it's needed. And actually you get a real. I think I donate blood. And it's nowadays you get a text telling you that actually this blood went into a baby or this blood went to somebody else or your plasma was used. And so you get told where it gets utilised.
That's a really good motivator. I love that.
Yeah, so I think it really helps. But there's an altruistic. I think British people are quite altruistic and they want to help out. That's what amazed me when we set out to do the clinical trial for Restore. I was like, will we convince people to volunteer to take this blood? When we injected the first two people originally volunteers. One was called Yvonne And she came onto the BBC with me, and I talked to her, and I had a chance to ask her why she did it and things like that. And that was really fascinating. You know, she had suffered breast cancer, and she got treated, and she wanted to give something back.
Yeah.
So that was her motivation. And that's just one person, and there's probably a lot of stories within the group of people. So.
I have a final question for you, each of you, or sort of cluster of questions. I started by asking you, where did you come from? What did you do before this? And you both started at your undergraduate days. If that Jan could sort of have a little window through time and see what you're doing now, what would she think? What would she say?
She would be very, very surprised, actually. Yes. Well, come from a sort of a maybe not a classical academic background as well. And the school I went to doesn't actually exist anymore because it wasn't the greatest school. So yes, I would have been very surprised. But I think my whole life has just been taking opportunities. I've not planned my life. I've not planned to go to this research lab or work my way through. It's been very much, this is what I fancy doing at this moment, so I'll apply for these jobs and go through. So yes, I couldn't talk to an undergraduate and say, this is how you plan your career. No. It's been very much like...
say that they're probably fibbing anyway, to be perfectly honest.
So it's been very much, this is an opportunity. I'll go with that and throw myself into it.
So how would, I guess, 18, 20-year-old Jan feel about you modelling your career like that? Would she find that surprising, or would that sound...
Wouldn't have understood how to model a career like that at all, really. Or have even known the possibilities, really. And I think that's an important one for school pupils in maybe less privileged areas, is to know that what the opportunities are and that they can do it. You know, you can do it. You don't have to.
You don't necessarily need a plan. You know,
you don't need a plan.
If you have a plan, that's great. But you don't have to.
And your plan will change though. I think if you have too strict a plan and it goes wrong, it can really knock you back. So I think just being open to the opportunities and taking them, and to believe you can do it as much as the next person.
And Ash, same question to you, whether it's schoolboy Ash in South Wales, whether it's undergraduate Ash here in Bristol.
I think they look and go, I can't believe you got to there, right? So hopefully they'd be really, like I would be really proud if it was me. But also I think they'd be like, how did you get to that point from just going to university and then going on? And like with Jan, it's just going from opportunity to opportunity and having the drive to do it. And I think the key thing is to take risks. And I think that's the same for entrepreneurship as well, is you have to take risks with science to do new things and try things out. But also you need the right people around you. So academics don't do this on their own, right? What I realised was I'm a zookeeper. So I'm in the zoo and all these people come in, all these amazing different people with different abilities. And they've all got different qualities. And then my job is to corral them and get useful things out of them. And because I've always wondered, like, I actually am quite intimidated at how amazingly smart people are when they come to university, and they're even better and better as you go on. So my job is to see what somebody could do, know that they can be pushed a bit more and encouraged and enthusiasm, and then they'll get to something that they never thought they'd reach.
Brilliant. Wonderful.
And then I think that's kind of what happened to me, right? So like I went from, not a great background. Like I was fine, but it was never privileged. And now I guess I'm middle class. I don't know what I am, but I've come from somewhere and gone somewhere, and I'm proud of my roots. But I'm also now a Bristolian because I've been here for 34 years or something. So
longer than you were.
And with your Scarlet journey, it's been not always what you initially planned. You've had setbacks, you've had challenges, but all said and done, would you do it again?
Oh yeah, definitely. Instantly. Yeah, it's good fun. So I am going to do it again. So probably I've always jokingly said I've got 3 companies in me.
Okay.
Because that's as far as my brain will stretch at the moment. But like there is one that's been set up in Berlin, and that's on a white blood cell company. And that idea has not been taken by me. The idea was formed together with another academic. And that academic is set up in Berlin, and they're going to run with it with my blessing, because I realised I couldn't do everything, and I will help wherever I can. And then I think I've got another one in me, which we're sort of exploring at the moment. And this is why I've got my enterprise fellowship, because I'm kind of like, I've got a new thing that I want to kind of explore over the next year or so.
Fantastic, exciting. We don't have a huge portfolio of serial entrepreneurs in our academic staff base, so I'm looking forward to utilising you as my poster child in the years
I'm not
to
trying to be serial. Like just kind of like one thing leads to the next thing, right? So
but
now I know now, well, you kind of know the process.
Yeah.
Once you know the process, so one thing is seek out people who've done this before, to find out what Yeah, so that I've really been blessed by the people around us. This ecosystem is in Bristol, because you can go and find other founders and ask them questions, and most people will tell you, they might not tell you all their secrets, but they will give you advice. And we did that, right? We sought that out. And my job now is to help other people do that as well, is what I see. And I'm sure Jen would see the same if somebody came and asked, if you had a student who asked, or if it's another academic. So come and see us if you want to have a chat about anything.
The whole thing does seem more accessible now than at the beginning.
Yeah.
I love that. That's a good call to action. I hope you don't get inundated. Thank you both so much, Professor Jan Frayne, Professor Ash Toye. It's been a real pleasure speaking with you. I'm really grateful for your time.
Thank you. It's been a joy.
Thank you.